In an effort to view the dynamics of the occupancy of the core-circadian activator complexes in the genome-wide context, one study assessed all core clock proteins in the mouse liver. At PER1, the activators BMAL1 and clock bind in a cyclic manner at the promoter between the circadian time 0, CT0, and CT12, with maximal binding observed at CT8. In genome-wide analyses, clock and BMAL1 bind to more than 4,600 and 5,900 sites, respectively, corresponding to approximately 3,000 unique genes in the liver. At highly expressed target genes, clock BMAL1 DNA binding motifs are enriched for a tandem E-box motif. Native liver extracts show that clock BMAL1 bind to tandem E-box sites cooperatively, with higher affinity to more effectively compete with other E-box binding factors, such as USF1.
Okay, thank you very much. So, what about there? Any questions on what they are saying, what they are talking about, what kind of experiments they are talking about? So this is in relation, maybe it might help if you go back to page 171, no, not 171, 169. So this is continuing the paragraph on the genomic targets of circadian clock transcription factors, where they are investigating what is the binding of clock proteins in the genome. And this paragraph which you read is the binding of BMAL1 and clock at genomic loci. So look at it and see if there is anything you... Can you remind me how do they quantify that? How can they quantify the binding of these clock and BMAL in the genome? Do you remember?
Use a lot of cells.
A lot of cells, yeah, we saw that was very important. We're using what kind of techniques they are using? Yes, ChIP-seq, very nice. So they essentially link, they treat the samples with a specific chemical that will bind the proteins to the DNA. So clock and BMAL, if it is bound to DNA, they will have a very strong link, a covalent link between DNA and proteins. Then they cut the DNA, and essentially the DNA that is bound to clock BMAL will be protected from being cut. Then, using an antibody, they will purify only these bits of DNA that have clock and BMAL bound to them. And then after that, they extract the DNA from this clock BMAL bound fraction, and they will sequence and quantify how many fragments of each sequence they have. So at a time where clock BMAL is not very much bound, then very little DNA will be precipitated, will be purified. But at a time where clock and BMAL is bound highly, then a lot of DNA will be in the sample, and a lot of copies of the same sequence will be in the DNA. And then they can quantify how many of these each segments of DNA. Is that clear? Is that clear what they do? Yes. What about you? Is it clear? Not so clear. Not so clear. Okay. So yeah, this is slightly It is slightly complex. So you imagine I draw, this is a whole chromosome. That's a whole chromosome. And it's not at all drawn to scale, but at certain locations in the chromosome, you have clock and BMAL that are bound to their gene of interest. For example, here, this is the gene PER1, for example. Okay? So what they do first is they use a chemical that will make a very strong link between the protein and the DNA. Normally the proteins, they are not bound to DNA using covalent bond. Okay? But this will generate a very strong covalent bond between DNA and protein. And then they will essentially cut the DNA. They cut completely randomly. They don't really know where they cut, but they cut. So this means that all the DNA here will be cut. Small fragments of DNA will be generated. Small fragment of DNA will be generated. Like this. So you have a population of small DNA fragments. Some of them are bound to clock and BMAL. Because clock and BMAL is bound, or other transcription factors are bound, they will not be cut. They are protected from being cut. Then they will use an antibody. You need a good antibody, cotype. They will use an antibody that essentially will recognize clock or BMAL. Clock or BMAL. Clock or BMAL. Clock. So these antibodies, of course, they will recognize clock and BMAL. In addition, these antibodies, they are linked to a big, heavy bead of metal. It can be different. It can be plastic or whatever. Very big metallic or plastic beads. But what it means is it's very heavy. This is very heavy compared to these fragments of DNA which are very light. They are not bound to protein, they are not bound to antibody, and they are not bound to this very heavy bead. Essentially, these fragments will sort of fall down into your tube. And so this is the tube that your reaction tube. And then you have your clock and BMAL. 1, 2, 3, 4. Clock and BMAL here with the bound DNA. I will not draw the antibody and the beads. So because it's very heavy, it goes at the bottom of the tube. Then you extract only the DNA from it. You take the DNA, you purify the DNA, and essentially you have 5 small fragments of DNA. And the only thing you need to do is then check what is the sequence of this DNA fragment. And if you know the sequence, you know where it is in the genome, and you know that, for example, this fragment corresponds to the promoter of PER1. Okay? So this is how you do. And simply, if this tube that you have comes from many different cells, each cell will contribute their own DNA fragment into the mix. So if it's at a time where clock and BMAL is bound very strongly to DNA, then you will have a lot of the same fragment from many cells. And not only you can check what is the sequence, but how many of these fragments of the same sequence. For example, what you found in the sample is this fragment that has the same sequence, you can find it 1,000 times. There are 1,000 copies of this DNA fragment. And this one, there are maybe 10 or 10,000 copies or whatever. So this is how you can quantify. And if you have many fragments, so let's say this one is at a certain time point A, and this is at time point B, and in time point A, this fragment 1, 2, 3, 4, 5, you have millions of them, right? Not 5, you have millions of them. Then this is maybe 3,000 copies, you have maybe 2,000 copies, you have maybe 500 copies. Then in the sample B, you find fragments with the same sequence, but the amount is much less. Maybe this is 50, maybe this is 10, maybe this is 30, maybe this is 1, and maybe this is 100. Something like this. So if you have very big difference in the amount of each of the fragments, then you know it is because clock and BMAP was not very strongly bound to these fragments. And only a subfraction of these fragments were verified. Is that a bit clearer to how they do things?
Yeah.
Okay, yes?
Okay.
If you don't understand something, tell me, what part of, what you do not understand in this? What is the part where you have some hesitation? Don't be afraid. It's okay. It's a second year lecture, so you should not be afraid. Yeah, you know, these guys are very strong. You don't need to worry. You don't need to worry. What part of this is difficult?何が難しい?
No, it's clear.
It is clear now? Yes? Okay. So this is what they do, okay? Of course, because of biological variations, clock and BMAL may bind very weakly to some region of the DNA. And you will get the DNA fragments that are bound to clock BMAL, but if it's very weak, you will have very little copies, 1 or 2, 3. And you know that even though BMAL1 can bind to these fragments, it's very unlikely. So these are not so very good target of clock BMAL. Okay, so this is usually what happens. All right. Anything else? What about this? What about this tandem mention? Clock and BMAL1 DNA binding motif are enriched for a tandem E-box motif. Repeats, yes. Tandem means, I told you that the E-box is the circadian E-box, CACGTG, is bound by clock and BMAL. But it's not only CACGTG. What they report, and some other people have said the same thing, is that only one CACGTG is not enough for very strong response to clock and BMAL. You need to have two, CACGTG, then some nucleotides, and CACGTG. So it's a tandem of E-box.
Figure 3A. So the first, this the amount of DNA collected.
Yeah. So I'll explain the figure 3A. Figure 3A. 3A, ne? Okay. So the figure 3, if you look at the figure 3A, I will explain what is the x-axis and what is the y-axis. So first of all, let me know what you understand and what you do not understand. So For example, I will only write here the data for B-1 because it's the same for the other one. I will write. So what you have is 0, 4, 8, 12, 16, 20. So this is time points, right? And then, of course, this is the data just for clock B-man. And then for each, I will write it in blue. For each of these time points, they show a blue line like this. And this is what the gene per one. Is this per one? Chromosome 1. Which gene is it? Which one they have chosen?
Chromosome 11?
Yeah, it's chromosome per one. This is chromosome 11. So this is the per one gene. Okay? This is the per one gene. And then here at the bottom of the figure, so you need to look below Cry2 in pink, you have this kind of a funny graphs like this. You have something like this, right? And then here you have many blue things like that. Do you see what I mean? Do you see what I mean? This line here, at the very bottom of the figure. It's written UCSC gene, UCSC gene. And then in addition to that, you have then the data where you have something like this, and of course it's lower, it's higher during the day, and it sort of decreases high, and then you have a little bit high as well here, but essentially okay. So this is a graph. There is a x and there is a y-axis. What is the x-axis? What do you think is the x-axis of this graph?
Sequence.
Yes, sequence. That's true. That's very true. The axis is the sequence, but the sequence of what? This is the sequence of what, the x-axis?
The place at which the clock and B-man binds, the place at which the complex binds to.
Yeah, this is the sequence that shows some binding of clock B-man, but not everywhere, okay? Not everywhere, not everywhere. Not everywhere. X is not really the sequence that is bound to clock B-man. What is the X sequence? What is the sequence of what? This is the sequence of something. This is certainly not the whole sequence of the mouse genome, right? This is not the whole genomic sequence. This is the sequence of a very specific thing.
Around gene.
Yes, this is the sequence around gene. Which gene? Per one, yes. So the x-axis from the beginning of the figure, just after the letter 0, 4, the beginning here, this is actually the beginning of the per one gene. And at the end here, this is the end of the per one gene. And what is this UCSC line that you have at the bottom? You know, you have on the between UCSC gene and the blue line, you have this kind of symbol, right? Like this. And it's written per one. So this indicates that this is the per one gene. It's written here. Per one. And here you have UCSC. So this is the per one gene. But what are these blue bars and thin blue line in this section?
The box is the other, and after that is the extra and inside.
Yes.
The two first places.
These big blue bars, these are exons. They are exons of Per1. My X is a little bit funny, it looks dead. Exon. These blue bars are exons, and this thin blue line, these are introns, okay, of the Per1 gene. It's DNA, right? So this is why you have introns there, because it's DNA. So this is exon 1, intron 1, exon 2, intron 2, exon 3, 4, 5, 6. And there are more exons than that. I cannot draw all of them. So, okay, this is the Per1 gene with its exon and its introns. Maybe I should draw it a bit more accurately, something like this. All right. So the x-axis is only a graphical representation of the Per1 gene with the sequence. Okay, this is the sequence of the Per1 gene. Now, 0, 4, 8, 12, 16, 20. This is easy. What is it?
Time.
This is time. CT0, 4, 8, 16, 12, 20. Which means this is the data from samples taken at different times. Of course, it's from a different mouse. Okay, it's from a different mouse because the mouse had to be killed every time point. But so this big y-axis is time. But what is the y-axis? So here the x-axis is the same. You know, it's just where we are in the genome in the Per1 gene. But what is? We have some sort of mountains here, a little bit here, and we have big mountains here, and these changes, they change with time. But what is this y-axis here? Actually, the scale is the same. The scale is the same, but what is the y-axis? From my previous explanation, can you somehow intuitively tell me what is the y-axis there?
Strength of mind. Strength of mind.
It's?
Strength of mind.
It is true. It represents strength of binding. In this case, I think of clock. P-1. But not exactly strength of binding. It represents strength of binding.
Number of sequences.
Yes, exactly. So this y-axis is the amount of copies of that sequence. So you said very earlier on that it's the sequence. It's true, it's the sequence. So what we have here is the amount of copies of each sequence that they detected. And the more copies you have, the higher the mountain, the higher the peak. So here, for example, what is the difference between the one in the promoter of Per1, before the first exon? You have this big mountain here, and it changes with time. And here, in an intronic sequence, you have mountains, but they also change with time, but they seem very small compared to this. What is this? What is the difference between this and this? Of course, you know that since it's two different places in the x-axis, they represent two different regions in the PER1 gene. This is the promoter, and this is intronic sequence. So that's all right. But why these peaks are very tall, which means that there is a lot of copies detected there, and these peaks are very small, so it means that there is not so many copies quantified. Why is that? What is the difference between, why is this very big and why is this very small? Speaking about the height of the peak, speaking about the y-axis dimension.
Email doesn't bind astronomy to the...
Yeah, so this means here that, so I told you the y-axis represents the amount of copies of each of these small sequence tags. This means that there is a lot of sequencing, there is a lot of copies. So in almost every cell, clock and BMAL was bound to this area at this time point. But here, this is significant. We can still see a peak, but somehow the binding of clock BMAL at that area was not so very strong. In some cells, clock and BMAL was bound, but maybe not in every cell, because the binding is not so strong. So then, the height of the peak was much lower. So which means that this is a site for high affinity binding of clock and BMAL, for BMAL1, and this is a site of low affinity binding. This is what it means. Is that clear?
And what does the mammal cons mean at the bottom?
What is the?
Mammal cons at the very bottom.
Aha. What could it mean? So, yeah, the green, that's a very good question. The last green line shows something in mammal. Cons means conservation.
Oh, so is it the general, the standard kind of pattern?
I'm not sure what you mean.
It's the genes conserved, so
Yes.
it's which are the same.
Yes. So the last line at the bottom shows if it's high, it means it's highly conserved. So if you compare the mouse, the rat, human, elephant, otter, whales, whatever mammals, then they will be very conserved area. So the local sequence, when you have a very tall, like for example, in the area where you have this big blue peak, you also have at the very bottom, I don't have green marker, that makes things easier. No, I don't have green marker. But at this location, you have a tall green peak here as well, right? Yes, you do. Yeah, it doesn't work. It doesn't work. So you have a tall green bar. There. This is what you have, right? You have a big green bar there. What does it mean? What is the meaning of a green bar there?
So does it mean that it's highly conserved, so that implies that it will bind like that?
Yes, exactly. So they mean this, not only this is a very significant binding site for clock BMAL, but among many mammalian per one gene, the sequence at this location is highly conserved, which means that there is a pressure to keep the sequence the same, because it is very important for the binding of clock and beam. So it means in mice and humans and rats and monkeys, the mechanism is the same. Okay, this is what it means. And of course, the explanation stands for BMAL1 and so BMAL1. What is H3K4 ME1? And H3K4 ME3. So this is figure 3a. Ne? So they have BMAL1, H3K4 metyl 1, H3K4 metyl 3, H3K9 AC, K27 AC, 6ME, and ME2. Whoa, what the hell is this? We said that before.
Where did you mention?
I think I jumped. I missed. I'm sorry. I am in the next figure. I apologize. So, yes, sorry, the figure 3a is BMAL1, clock per 1, per 2, Cry1, and Cry2. But looking at figure 3a, so this is the explanation for BMAL1. If you take a look at the Cry1 binding site, you can see that it's located at the same region. The binding site of Cry1 is actually the same as the binding site of BMAL1 and clock. But what is different? If you look at the y-axis, without reading the text, what can you see?
Peaks or like the level. So there's two of them.
Yeah, it's not exactly the same, right? Like for clock you have something like this, and for cry you have something that is a bit more equal like this. That's one difference. It's not that significant, but means they bind to the same location. Maybe not exactly with the same affinity here and there, but they bind. But what else can you see in terms of differences between the Cry1 binding and the BMAL1 binding?
The time they reach the highest affinity.
Yes, the time of high binding is a little bit different. For example, if you check for Cry1, CT20, the binding is very high. But CT20 for BMAL, it's not so high, right? So of course, this is related to the fact that they have different rhythm of expression. So when BMAL and clock is high, this means that induction of EBOX regulated genes. But when Cry is high, then BMAL1 and clock, they are inhibited and genes are not transcribed. So of course, the binding of Cry and BMAL1 will be slightly different. But it's difficult to say. Now maybe for Cry2, it's a little bit clearer that when Cry2 binding is high, BMAL1 binding is low. But you cannot really say the same things for maybe, for clock. Of course, clock looks like BMAL1. Per1 is also opposite to what you observe for BMAL1. Per2 is also opposite. So Per2 essentially, the binding of Per1 and Per2 is highest at night, while it is lowest for BMAL1. So you have this kind of relationship between the two. Cry1 is a little bit less clear, but you have some tendency. Is that okay? Figure 3a. Yes?
What is the function of the low affinity binding site?
What is the function of?
The low affinity binding site. This small bit here. That's a very good question. I mean,
what is the function of this binding site? And what is the function of this binding site? The question simply is, we do not know. They may just be additive. For maximal regulation of PER1, maybe you need this binding site and this binding site. This is possible. What could be the role of this binding site? I'm just asking you to give hypothesis. We don't know. We don't know. What could be the binding site? What could be the binding site here and the binding site here? What could be potentially functions that you may want to investigate when you come to our lab and study? What might be the function of it? There are different binding sites, apparently of different affinities, and they are in different regions of the PER1 gene. So what could there be? What could be the function of these two sites?
Transcription, maybe? If they increase transcription.
Yeah, but this could be the same also for this one, right? So you could say they are both important for transcription activation of PER1. This is true. Or they are important for clock B-mal regulation of PER1. This is true. But what might be different?
Like in a very special type of cells, that's the actual the important transcription.
That's very possible. So what we see here is data from the liver. So maybe from liver samples, clock and B-mal is bound very strongly here, especially during the day. But in the brain, for example, or the kidney or whatever, then this binding site may suddenly become stronger. That is possible. What else?
If it's spliced differently, maybe?
Yeah, that's very good. So, what if, yeah, you have PER1 gene, but PER1 gene is only one gene, but it can be spliced differently. One gene can maybe give 2 or 3 different PER1 mRNA coding for different PER1 protein. So what if there is a different messenger RNA that does not contain this exon, but starts, let's say, from this exon? So you have a much smaller PER1 protein with a different sequence whose promoter is here. And this is where clock and B-mal bind to promote transcription of this PER1, smaller PER1 protein. That may be one of the cases. That may be differential regulation of different alternatively spliced messenger RNA. That may be a very possible explanation. And actually, it might be very true because PER1 indeed has many different messenger RNA, and it has never been confirmed. The function is not known. The function of this messenger RNA is not known. But it seems indeed that PER1 has different RNA. So you could. Or it might be simply that these introns are very big, right? They are very long. So this binding site is very strong of clock and B-mal. So of what we saw before, this clock and B-mal will also change histone structure and will promote transcription. But then polymerase will start transcribing the messenger RNA, but it's quite a long way away from the original activation here. So what could be this role here is a kind of reactivation of the RNA polymerase. It's just telling the polymerase, hey, you have to continue, you have to go faster. It's very long, I know it's a long way away from where you started, but you need to keep on going. It's a kind of, hey, remember, you have to activate transcription. And it doesn't need to be so high because it's mainly where here it starts, but it's just an, hey, remember, you have to transcribe. That's maybe also the case. But it's only possibilities. Nothing has been confirmed. All right. Then we are on page 172. If my memory is correct. Where is 172? 173 then.そうだね。The hepatic steroid あれそうだね。The hepatic steroid receptor co-activated ね。これね。そう。どうぞ。
The hepatic steroid receptor coactivator 2, SRC2, also known as NCoA2, is recruited to DNA in a diurnal cycle and overlaps extensively with CLOCK-BMAL1 sites. SRC2 acts as a potent transcriptional coactivator of CLOCK-BMAL1, and null SRC2 mutations in mice lead to an abnormally early phase of the activity rhythm, indicating that SRC2 has a role within the circadian clock system.
Okay, that's okay. Thank you very much. So this is a little bit of additional information. It's not very important, but essentially it says there are many things that contribute to the activation of genes by CLOCK-BMAL. Anything you do not understand in this paragraph? What is a null SRC2 mutation? Just, it doesn't function. It's a, knockout mutation. Okay, then we can directly go to the next paragraph.
In addition to the liver, the circadian system has recently been described in mouse pancreatic islet. In the comparison of the occupancy of BMAL1 between the liver and pancreas, about 40% of the target genes overlap with disparate binding site clustering and distal-regulated regions of tissue-specific genes. Interestingly, even within the overlapping set of target genes, BMAL1 was found to bind to distinct enhancers in the liver and pancreas, consistent with the existence of tissue-specific circadian cis-regulatory modules.
Okay, thank you very much. So what about this one? Pancreatic islet is a Swiss name. It's in the pancreas.
Islets of Langerhans?
Hmm?
Does it mean the islets of Langerhans?
Yeah. So, is that okay, this paragraph? What does it tell us? This is about the difference between tissues. What does it tell us with your own words?
In different organs, the CLOCK-BMAL complex binds to different quite different place and enhancer or different genes have different sequences.
Yeah, exactly. Well, it simply means that there is only about 40% of common target genes between the liver and the pancreas. So which means that there are 60% of genes that are different. They are target of ClockBMAL, but in a tissue-specific manner. And it's not only that, but if you check the genes that are targets of clock and BMAL binding, clock and BMAL binds to different regions in these genes. And this is almost similar to what we said before. As you said before, that indeed these bindings may be different. This one may be the main one in the liver, and this one may be the main one in pancreatic islet. Which means it may also be in conjunctions with what we say, is that in the liver, the main PER1 transcript is the one that contains all the exons here, and that is activated by this one. And in the pancreas, we may have a smaller PER1 transcript that is using this side. We don't know. But what they say is that it's really different. The binding of ClockBMAL is different, not only in different genes, but within the same gene in different locations. But not always. Not always. Some bindings are the same. Some bindings are the same. All right, then we can go to the next paragraph.
Genomic targets of the core circadian repressors. In contrast to the activators, the repressors PER1, PER2, and CRY2 bind genome-wide at night between CT12 and CT20, peaking between CT15 and CT17. CRY1 has a bimodal pattern, with a major peak at CT0. The repressors CRY1 and CRY2 bind to significantly more sites in the genome than PER1 and PER2, with many thousands of these sites being independent of ClockBMAL1 and containing DNA binding motifs for nuclear receptors. Consistent with this finding, CRY1 interacts with several nuclear receptors and is a co-repressor for the glucocorticoid receptor. The nuclear receptors REV-ERB alpha and REV-ERB beta also occupy DNA rhythmically, with a peak in the daytime. REV-ERB alpha competes with ROR alpha for DNA binding at RORE and REV-DR2 sites to regulate circadian target genes such as BMAL1 and CRY1, where it recruits nuclear receptor co-repressor 1, NCOR, and HDAC3 to repress gene expression. Interestingly, REV-ERB alpha also interacts at other genomic sites that are enriched for tissue-specific transcription factors, such as hepatocyte nuclear factor 6 in the liver. At these sites, the action of REV-ERB alpha is independent of its DNA binding domain and defines a different set of target genes that are directly involved in metabolism. Finally, SRC2 has been proposed to modulate an oscillation in chromatin accessibility by recruiting PVAF members of the SWISNF complex to promote REV-ERB loading and enhance the amplitude of circadian gene expression. Thus, the circadian system is highly dynamic and involves the core circadian EBOX transcriptional regulators and is interlinked by REV-ERB alpha beta as well as EVP and NFIL3 to secondary and tertiary gene targets with a wide range of phases of gene expression.
Okay, so this is essentially a very complex picture of what we saw earlier. In a way, we have the clock and BMAL loop, the PER1 CRY1 loop, the DBP and FIL3 loop, the reverb alpha ROR loop. Everything regulates everything. It's really interlinked feedback loops. And as they say, so CRY1 is a repressor of clock and BMAL, but not only, because CRY1 can also interact with nuclear receptors and also co-regulate gene expression that are targets of these nuclear receptors and CRY1. So there is a lot of simultaneous regulations, which essentially makes the clock a very, very strong molecular mechanism with many layers of regulation. An advice for the future these gene names are very difficult, and in the circadian field, we never say REV. It's so difficult, so essentially we just say reverb alpha, reverb beta. We say rho alpha, rho beta, as you said. So sometimes just say the HN. Yeah, it's sometimes HNF6 is still HNF6, so it's a little bit. HDAC3. We say HDAC3. Oh, okay. Any questions for this long paragraph? Any specific? So I see they tell us now that not only reverb alpha and ROR alpha, they bind to RORE, but also to rev DR2 sites. And it's just different sites. Slightly different sequence, but the structure of the site is different. But in some cases, it has been found that these different sites can also be bound to ROR and reverb. Two different sequences, but still binding site.
What's the glucocorticoid receptor?
What is the glucocorticoid receptor?
It's come up in another lecture, but I've got that.
What is the glucocorticoid receptor? Receptor. What? Where is the receptor? Where is the receptor?
It's not hormone, is it?
Glucocorticoids are hormones. They come from the adrenal gland. They come from the adrenal gland usually. But where is the glucocorticoid receptor?
It's in the surface membrane.
Is it?
Inside the cytosol.
The cytosol. Where is it?
Liver.
Yeah?
Liver.
It's everywhere. It's everywhere.
Everywhere.
It's almost everywhere. Many tissues respond to glucocorticoid hormones. So essentially, the glucocorticoid receptor, it's a transcription factor that when it binds the glucocorticoid receptor, actually regulates gene expression. So it's not a cell membrane receptor. It's an intracellular receptor that is regulated by binding to glucocorticoid and then regulate gene expression by going into the nucleus and activating there. So then it can interact with cry and these things because it acts in the nucleus.
Is it in response to cortisol?
Yes, for example. One of the did I explain that before? So when in the laboratory, when we do, it's a little bit outside of the paper, but since you asked the question, it's interesting to mention now. I've maybe already explained something similar, but so let's imagine you have a population of mice. Say you have 18 mice, and you kill 3 mice every 4 hours. So you have CT0, CT4, CT8, CT12, CT16, and CT20. And here you kill 3 mice, 3 mice, 3 mice, 3 mice, 3 mice, and 3 mice. You extract the liver, and you quantify per 1 gene expression or per 2, and what you see is something. And that's, let's say, per 2 expression. But if you take a cell culture, you take cells in the dish, and you measure per 2, and you measure at different time. Doesn't matter, it's not really CT in cells because cells has no locomotor activity, but let's just say it's every 4 hours. And you get flat line. if you maybe remember what we said before, what is the conclusion? This is PER2. This is PER2. And let's say you took these cells from the liver. They originate from the liver, so you know PER2 is expressed, but somehow, when you measure the expression of PER2 in a cell culture dish, it's completely flat. So what is the conclusion you draw from that experiment regarding the clock in the cells? That you get a rhythm of PER2 when you study the PER2 expression in the liver, but when you measure PER2 expression in cells, it's completely flat. What conclusions could you draw from this experiment?
That maybe it's not cell, it's not just the cell that's regulating it.
Yes, maybe you need to have a tissue context to get regulation of PER2. This is possible. Maybe you need the liver connected to the brain to have rhythms. However, if you don't take cells, but you take a big piece of the liver, you put into a dish, you cultivate, directly after preparation you start sampling, and then you get rhythm. So tissues taken out of the body can happily continue their rhythms, even in a dish. But somehow, when you get single cells,
Don't the single cells spread flat?
yeah, they are evenly distributed in the dish. Can you say that in vitro the cells have lost the clock? Can you say these cells are not oscillating? Yeah, I mean, you can certainly say that quantifying population-wide PER2 expression reveal no rhythm in PER2 expression. This is exactly what you see. Okay, so then what you do is saying, all right, so maybe, as you said, maybe in the body, maybe some signal is necessary to keep the clock ticking. And if you are in a dish, maybe cells, because they are single cells and separated, they don't receive these signals. Maybe when you take a big tissue block, still, maybe something happens. So what you want to do is treat these cells with hormone. And say, for example, I'm going to add into the dish a very powerful hormone that many cells can respond to, glucocorticoids. And then you use glucocorticoids. You do the same experiment, but before sampling, at -2 hours, you add glucocorticoid. And the name of the glucocorticoid you use is dexamethasone. And you treat your cells for 2 hours. After that, you remove dexamethasone because you don't want dexamethasone to always be there in case it does something not nice to the cells. And then you measure PER2 expression and you get like this. That's exaggerated. You can have that for many days, but eventually you will get a flat line. So, what's the conclusion? These cells have the ability to oscillate after being shocked with dexamethasone. And so this is where you see that glucocorticoid receptor definitely has an effect on the clock. But this is only temporary. After some time, you get flat. But then when it gets flat, if the cells are still alive, because it's a long time in a dish, if you give glucocorticoids, they will again start to oscillate. So what's the conclusion of that experiment?
The cells kind of oscillate independently but require the glucocorticoids to
Yes, they can oscillate, but you can also answer. It's not. The cell can oscillate, but they require glucocorticoid. Does it mean that glucocorticoid will start the clock? The clock is not working. The clock is broken. But when you add glucocorticoid for 2 hours, the clock starts. But after a few days, again the clock will stop. Is it what's happening, or is there another explanation?
It's only necessary for the transcription of parts. No.
Yes, it could be only necessary. Maybe the presence of dexamethasone will activate glucocorticoid and glucocorticoid receptor, and only PER2 will start oscillating. But maybe if you check BMAL1 or CRY, you will see oscillations. Actually, no, it's the same. If you check gene expression, they will all be like this. After glucocorticoid, you have some oscillations, but they will eventually decrease. So the first possibility is that glucocorticoid is necessary to start the clock, but then after some time, the clock of the cell stops, and you need again provide glucocorticoid. Almost like having a watch, this old watch that you need to rewind every now and then. Maybe maybe dexamethasone is a kind of rewinding the clock, or is there another? Keep in mind that here we are quantifying clock genes, CRY1, in this case it's PER2, but it's the same with BMAL1, CRY, CRY2. We are measuring the whole cell population. We have cells in a dish, we take all the cells, we extract RNA, and we quantify what is the level of clock gene messenger RNA. And if you forget about biology, and if you think about physics and oscillatory physics, there are at least 2 different reasons for this kind of pattern.
The cells are oscillating at different phases?
Yeah, exactly. So this is actually exactly what happens. So in a cell population, every single cell is a little bit different. And the period of each cell is different. So it means after dexamethasone shock, this resets the cells, everybody at the same time, blah, time 0. And then after dexamethasone, I will write only 3 cells in each column. After dexamethasone shock, it's a very strong resetting, so all cells will start at the same point, like this, and cell 1 will go like this, cell 2 will go from the same point like this, And cell 3 will go like this. That's a little bit exaggerated, but in the beginning they are not so different, but because they have different period length, they progressively go out of phase with each other. So if you calculate the average of each cell, what you have, great, it's a great marker, it doesn't work. If you actually measure the average, you have one single curve. You have one single curve. In the beginning, all the cells are in phase, but progressively, they sort of cancel each other out. And so this is finally the point that I wanted to make. Glucocorticoid has an effect on the clock. The strongest effect is the synchronizing. All the cells get synchronized at the same time. And this is used in cell culture to make sure that all the cells are at the same time. Because if you don't do that, and you, for example, quantify what they are doing there, if you quantify the binding of clock in cell culture at different times, you must make sure that your cells are all synchronized. Otherwise, you never see any difference. Because they all have different phases. Okay, so that's it. Any question? No? Really, don't hesitate. Okay, then I will read. Oh, maybe, okay. We have reached the end of this paragraph. So let's start next time at genome-wide circadian RNA expression. There is 20 minutes left. Do you want to continue or do you want to stop early today? Yeah? Let's continue? Or that's enough?
It's okay.
It's okay? Okay. It's okay means let's stop. Or it's okay is let's go. Okay, then I will continue to read this one and then we can stop. Genome-wide circadian RNA expression. To examine the functional consequences of the circadian system, RNA sequencing, RNA-seq, has been used to profile cycling genes in the mouse liver. For example, using both the intron RNA signal as a proxy for pre-messenger RNA and the exon RNA signal as a proxy for messenger RNA, strand-specific whole transcriptome RNA-seq revealed a robust circadian oscillation at the DBP and HER2 genes. As was previously observed in microarray experiments, thousands of genes cycle at the steady-state messenger RNA level in the liver as well as in many other tissues. As more regions in the mouse have been profiled, more than half of all genes have been found to cycle at the messenger RNA level in at least one tissue. Importantly, the identified cycling genes are enriched for drug targets. More than half of the top-selling 100 drugs on the market in the United States have a circadian target gene. This finding emphasizes the importance of optimizing the timing of drug administration for circadian drug targets, which can both enhance efficacy while at the same time reduce side effects. Okay. And here, he is again citing his work. And there is one mention that I want to make, is the definition of most of these scientists, how they, when they say cycling genes, how do they define cycling genes? And I think I disagree with many of these scientists. maybe it's a bit early, but let's just see. Let's say you measure something and you have two conditions. You have conditions, so you are measuring, for example, the amount of per2 protein concentration. And you measure two conditions, A and B. And you have condition A, there is that much protein, and condition B, there is that much protein. For each condition, you selected 3 replicated samples. So you have 3 samples, and you have 3 samples for each condition. So you don't measure only 1 sample for each condition. You have 3 samples. So that means for each of the sample, you have a value. Let's say for this one, we have a value of 2. And this one, we have a value of 3. But that is the average value. Do you know average? Yeah? Mean. You know mean. So that means for the 3 samples, maybe if I draw in blue, the value for each of the 3 samples at condition B, maybe the sample 1 was here, the sample 2 was here, and the sample 3 was here. So you calculate the average, you get 3. And for number 2, for the condition A, again you had 3 samples, you had 1 sample here, you had 1 sample here, and you had 1 sample here. So that's the average. So what you want to know now is the difference between A and B. Is it significant? What you see, does it mean something? So what I am talking now is about statistically significant. It looks like there is a difference, but is it statistically significant? Do you know that already?
I'm studying now.
You are studying now. What about you? You know. So what kind of tests, in this case, what kind of tests could you use to know whether it is statistically significant?
Chi power 2.
That's much too complicated. You have 2 conditions. 2 conditions, A and B, and you want to know whether A is significant to B. Now it doesn't matter. You do this in this case. You do student t-test. That's a very easy, one of the easiest statistical tests. Student t-test. What about having 6 time points? Not only you have A, but you have CT4, CT0, CT4, CT8, CT12, CT16, CT20. And you have different levels. What is the test you use to know whether this is different from this, 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 and this is different from this? ANOVA, yes. It's if you don't know, it's okay. It's called ANOVA. So this is just like t-test, but you analyze more than 2 samples. And you have a general significance when it tells you there is a difference in time among these samples. And then after doing the ANOVA, you do another more refined test that will tell you that maybe this is different from this one, but this is not different. This is different from this one, but these 2 are not significant. So anyway, you have something like this, and you know which is different from which. This is very good. This is nice. But the test that these guys are using to know whether it's rhythmic or not is completely different. The test they are using does not take into account the significance between the data. They only take a look at the waveform of the thing. So let's say if you have, so this is 1 here, this is 2, and this is 3. This is just the amount of messenger RNA, for example. And imagine you have something like this. You have something like this. And they will tell you, there is a rhythm. But actually, if you do that, if you analyze that, so you have 0, 4, 8, 12, 16, 20, they will tell you that there is a waveform, but they have never checked whether there is statistical significance between each of the points. And they say that this is oscillations. Now, I am sorry, but there is no difference. Maybe there is an oscillatory pattern, but if you don't ask whether this is significant from that, whether this is significant from that, this from that, it's meaningless. And if you actually check the real data they're actually using, many of the genes that say they are rhythmic, they look like that. Now, it is very different. It's okay to do this test to know whether you have a rhythm, but first you must do an ANOVA. So if you have a gene like this, like for example PER1 will be like this. Completely different. And of course, after an ANOVA, this point will be very different from this one, will be very different from this one. The statistical significance will be different. And after that, once you have statistical significance, then you can check whether your data describe a waveform pattern. They usually skip the significance part, and they only check whether there is a significance. I don't think it's so good. I don't think it is not very good, because if you have a little bit of noise in your data, a little bit of variability, this will induce some rhythm. So anyway, usually they are making the data much better than they are actually. It's true that there are many genes that have rhythms, but maybe not as much as what they say. Okay? But the point is true. There are many genes that are regulated by the clock that are rhythmic. Many of these genes are important for some disease or others. They are different between tissues. Each tissues have different rhythms, but rhythms is not everywhere. There are genes that are completely flat. Okay. That's also exists. Anyway, that's the end for today.